Every year, 45% of managed honeybee colonies die in the United States. The reason? Varroa mites, along with a bunch of other factors. These red-brown parasites attach themselves to adult bees and larvae in the bee colonies. They then feed on these living bees, devouring them from the inside out. If not taken care of, nearly all bee colonies will be eradicated, and we wouldn't have the variety of food that we have today. No coffee on your way to work, bread for sandwiches, or avocado for toast. Without honeybees, our plates would look very bare.
Honeybees play a vital role in U.S. agriculture. If we were to break it down, honeybees produced 148 million pounds of honey in the United States in 2020. With the price of honey being $2.03 per pound, honeybees were estimated to add a little over $300 million to our economy. That is just the tip of the iceberg, however. Not only do these creatures produce honey, they also produce 5 other hive products: pollen, royal jelly, beeswax, propolis, and venom, all of which are very important to our economy. In total, honeybees are estimated to add around $15 billion to various sectors such as agriculture as well as healthcare and consumer goods. Taking care of these tiny but impactful creatures should be of interest, because if we don't, we also have to imagine a world without apples, berries, vegetables, candles, cosmetics, and even certain medicine that we take for granted every single day.
Despite their importance in sustaining our daily habits and way of life, honeybees are constantly neglected for the incredible value they provide. Issues that affect us as humans also affect this buzzing insect. Deforestation, climate change, and high pesticide use all contribute to the death of millions of bees each year, conveniently named the colony collapse disorder. But with breeding tactics, beekeepers are generally able to revive entire colonies and prepare for the next pollination season. One predator, however, that doesn't just affect current bees, but can alter traits and impact future generations, is the varroa mite. The varroa mite is a pest for bees and beekeepers alike. During pollination, while a worker bee is perched on top of a flower, varroa mites will often jump on the bee's back undetected. The mite will use the worker bee as a transportation system, laying low until the bee returns to its colony, all the while waiting for the opportune moment to jump off and nestle itself into the beehive. This is where real disaster begins. Although these red-brown parasites feed on the fat of adult bees, they have a more devastating mission in mind: infect and weaken the babies within the colony. This means that these parasites aren't just annoying to kill, they have a keen sense and tend to seek out bees in the larvae and pupae stage within the developing brood of the hive, feeding and reproducing right in the heart of the bee colony. Varroa mites pass on diseases and weaken the developing bees. These bees often grow to be crippled and can only crawl, have impaired flight performance, lower productivity, reduced lifespan, and reduced weight. These infected bees might even spend more time in the hive, increasing the likelihood of parasite transmission within the colony, accelerating the spread of the infection. What's more is that these disorders are then passed on to future generations of the colony during reproduction, making them even weaker and impaired in their functions. All the while, the varroa mite remains hidden in the hive. If not addressed quickly, the bee colony will eventually produce useless bees that are prone to death without any way to revert this genetic disorder. This is where technology comes into play.
Back in 2004, the U.S. Department of Agriculture started its venture into genome sequencing for bees. Ours, or USDA's Agricultural Research Service, in collaboration with Baylor College of Medicine and U.S. National Institutes of Health, released a draft version of the honeybee genome to the public in January of 2004. The aim of this project was to better understand the position and order of genes residing on the insect's DNA, providing bee researchers with a shortcut to traits that can otherwise be difficult to identify. The USDA Undersecretary for Research, Education, and Economics, Joseph Jen, stated, "This research puts the honeybee center stage as the first agricultural animal that's been fully sequenced. As an organism whose social order rivals our own in many ways, the honeybee will serve as a natural system for further agricultural studies, including such areas as social behavior, cognition, and immune system function." Over time, the price of genome sequencing has decreased. Rather than costing thousands of dollars as it used to, honeybee sequencing now only costs around $100, making it more accessible for everyone, including beekeepers. As a result, beekeepers across the nation have started to take advantage of this advanced technology to better understand their hives. When a colony is sick, a beekeeper may choose to send over some larvae and bees to the USDA Bee Lab to conduct sequencing. Through this, beekeepers can understand the cause of sickness, whether it may be due to genetic mutations or pesticides.
This project is going to take those sick larvae and sequence everything in them, and basically get the whole soup of microbes in them. And that'll tell us, we hope, if which microbes tend to keep showing up in these sick larvae, and then maybe are causative. We can test them to see if they cause the disease. And then in some cases it might not find any, and then the bees might be sick because of pesticides or even nutrition.
This is Dr. Jay Evans, a research entomologist at the Bee Research Laboratory at the USDA, and one of two co-authors of the proposal to sequence honeybees in 2004.
Within a week, say, or less, okay, you've got to treat for bacteria because this is predominant. This is probably what's causing your issues. Or it's a virus and we don't really have good antiviral treatments yet, or we're searching for those. So in the case of a virus, we would recommend maybe just quarantine or somehow reducing the movement of bees. So that's one applied thing. The other one is to actually breed bees that are resistant to viruses and other disease. So there's a USDA lab in Louisiana that's specialized in that. And they're trying to use these genome tools and literally just resequencing genomes of resistant bees and finding out from that, okay, if we keep track of these 10 markers, we'll get half of the increased resistance of that line or something like trying to bolster it that way. And
what Dr. Jay Evans is talking about is selective breeding programs, a project with two goals in mind. 1, breeding specific hygienic traits, and 2, stopping mites from reproducing. Selective breeding is a process in which positive traits are identified and the bees which displayed those traits are artificially bred together. This way, the preferred traits are passed on from generation to generation. With the advancement of genome sequencing, selective breeding programs have simplified as traits can be identified in the genome sequence of a bee rather than having to physically test for them. Before genome sequencing, however, beekeepers and researchers manually engaged in selective breeding through a series of tests and experiments. In the 1980s, Dr. Marla Spivak and Gary Reuter started this for honeybees, attempting to increase the honeybee population with traits known as hygienic behavior. From weeks 2 to 3 of a honeybee's lifespan, bees engage in something known as hygienic behavior when removing corpses and infected eggs from the hive. This hygienic behavior is instilled in bees to prevent disease from spreading across the hive and wreaking havoc. To test the colony for hygienic behavior, the process would entail freeze-killing a portion of the brood and see how much of the dead brood the bees would remove. Obviously, the bees which would remove more of the dead eggs would have the strongest hygienic behavior. The bees with the strongest behaviors would be bred together, and a cycle of testing would continue repeatedly until the desired level of behavior is achieved. The selective breeding that both Dr. Marla Spivak and Gary Reuter did as a result of their research went on to become the infamous Minnesota Hygienic Line. The line has selected and bred bees carefully to eliminate the threat of certain diseases that stem from parasitic mites, malnutrition, and pesticides. Consequently, bees with such traits are in high demand. But there's one main problem. Breeding bees for specific traits and immunity removes genetic diversity that bees inherently have. As Dr. Marla Spivak explains, bees are known to be very diverse creatures that all have different traits, similar to humans. But by selecting certain bees to breed, this genetic diversity becomes lost. This contributes to the homogeneity of honeybees, which is bad because it limits the natural traits and genetics that bees would have developed through evolution. This is why Dr. Marla Spivak had to discontinue her Minnesota Hygienic Line in the late 1980s.
Beekeepers were really liking the hygienic line and asking for more and more queens. And I started to freak out a little bit because one of the most important traits for honeybees is genetic diversity. That trumps everything in colony health. That's the most important, is to make sure there's enough genetic diversity. So it's kind of like a Swiss Army knife, so that there's enough bees that can get all the tasks done. So I was afraid that if too many people in the US, beekeepers in the US, started using this line of bees, the genetic diversity would go down. We'd have a genetic bottleneck. And I thought that would be a disservice to bees themselves and to the beekeeping industry. So I just stopped and said, this is easy to breed for, do it yourselves.
As mentioned before, the second goal of bee genome sequencing is stopping mites from reproducing.
They can't reproduce anywhere else. They have to develop on honeybees, and they have a very narrow window when they can parasitize them and actually reproduce. And they pick that window by smell, so they identify when the bee is at exactly the right age that they can have time to reproduce on her. And so there's efforts to kind of look at the chemicals involved with that and block them somehow.
The ultimate question remains: can we save the bees? Absolutely. Researchers and scientists are making strides in creating solutions aimed at improving the health of bees. With the introduction of genome sequencing, beekeepers and scientists are able to create lines of bees with stronger traits that are less prone to diseases and illnesses. However, this is not a permanent solution to the problem. Illnesses and diseases continue to evolve at a rapid pace and get stronger with each iteration. Soon enough, they will become resistant to the hygienic behavior from breeding, and it will not be enough to save the bee population. Is it time for more technology to play a role in this industry? Watch part 2 of this series to learn how companies are creating in-hive solutions to address bee health.